Okay, according to my clock, it's 13:40, and hello, good morning, good afternoon to this first SOL International 2020, an online version. Now, I'll just give you some background. So the SOL meetings, for those of you who are not familiar with the SOL meetings and joining for the first time, the SOL meetings are held on a yearly basis until now, since about 16 years, since 2004, when the first meeting was held here in Wageningen. Unfortunately, due to the COVID pandemic, has been postponed. It will be held next year in Thessaloniki, in Greece, and hosted by Angelos. And so you're all very welcome to attend that. A co-chair group to discuss whether we do an online meeting. So that's what this is. We decided to call it SOL International. We will see about the success of this meeting, and if it's a big success, I think we'll have more of these online meetings in future. But we're very open to discuss whether we alternate between online and live meetings. Certainly the live meetings will continue. So the organization, well, as I said, the co-chair group, all of us helped in the organization. I have to give special thanks to Susie Strickler and to Aureliano Bombardet for their sterling work in helping to organize this. And I'm sure it's going to be a big success. For those of you who haven't got there yet, the book of abstracts and the program is available on solgenomics.net. Thanks to Lucas for posting all those things up. So we have now, right now, about nearly 200 participants. So welcome to all of you. We have 930 registrants from 50 countries, all continents except for Antarctica. And they come from a mix of places, industry and academia, of course, and also independent people have come along. So that's great. We will have about over 30 presentations in 6.5 poster presentations, which are available. The links are given in the chat. So each session, each of the 6 sessions has an invited speaker to kick off the session and 3 other submitted presentations. Welcome everybody very warmly, and I will transfer over to Giovanni Giuliani and Joyce Van Eck, who will host the first session on biotechnology, and it's over to you, Giovanni.
So, hello everybody, and welcome to this first session of SOL Online 2020. We have around 280 participants at the moment, growing quickly. This is the biotechnology session. We have 6 very good talks, an introductory talk by Ellen Goossens of Ghent University, and then 5 short talks. All of them are 4 short talks. All of them are very good quality. We were really in difficulty choosing the abstracts for the oral presentations. The way it will be organized, it will be that the first talk has 25 minutes, 20 for presenting and 5 for questions. And then the remaining talks have 15 minutes each, 12 for presenting and 3 for questions. We will alternate. Me and Joyce will alternate moderating the talks, and we'll come in just 2 minutes before the time for talking is up. So it will be at 18 minutes for the first talk and at 10 minutes for the remaining talks. The speakers, we have 2 minutes to wrap up and then time for questions. The questions have to be sent through the questions and answers button on the bottom of your screen. And they will read by the moderator in the order they are received. So don't use the chat, use the questions and answers button to send your questions for the talks. So without any more ado, I will introduce the first talk, Alain Goossens from Ghent University, and the title of his talk is Unraveling the Regulation of Tomato Metabolism through Interatomics Analysis. Alain, the floor is yours.
Okay. I will share my screen.
Okay, thanks. We have to move on. Please remember all the sessions are being recorded and will be visible again after the session is finished. So I leave the floor to Joyce, who will introduce the second speaker.
Okay, before I do the introduction, I'd like to remind everyone that after this session, there's a break, but we also have a Zoom meeting room. So if you want to, ask other questions of the speakers, you'll have that time to do that as well. And there are links in the abstract book for joining these Zoom rooms. Okay, so our next speaker is Peter Waterhouse. Peter is from Queensland University of Technology. And the title of his presentation is Genetic, Epigenetic, and Metabolomic Insights from Reference Level Genome Assembly of a Wild and a Laboratory Strain of Nicotiana benthamiana. Peter?
Can you hear me okay?
Yes, I can hear you fine.
Okay. Share my screen. And can you see that okay?
Not yet, Peter.
Oh, sorry. I hit the share button. How about that?
Oh, that looks good. And if it's in presentation mode, that'd be best.
I'm not sure how you do that in presentation. No, so,
okay,
oh,
I think it looks,
I'm still looking at the wrong screen. I'm looking. Is that better?
No, go back to where it was. Your presentation screen is in speaker mode. Okay, that looks good.
Okay. So I condensed the title. It was a bit long, so I turned it to the nature and nurture of Nicotiana benthamiana. And I just quickly acknowledge the people that have done this work. So there's my team in Australia, but it's in collaboration with Ori Bombali, Giovanni and his team in Italy, Diego, his team in Spain, Victor Lacca at Cortiva, and Chris Weinfeld in New Zealand. And so I'm sure I don't need to introduce very much what Nicotiana benthamiana is all about. It's a very good research tool as a plant in the research lab, and it's increasingly being used as a biofactory plant for things like vaccines and antibody production. So we've been sequencing the genome of the lab strain that everybody uses around the world, and it comes from this place in Central Australia. It's called the Granites. It's near a gold mine, and that was collected there in about the 1930s. So we've been sequencing the lab strain, but we've also been sequencing a wild strain from Queensland, which comes in this area here. These checkered regions are where Nicotiana benthamiana grows naturally in Australia. So just to give you a quick picture of the differences. So this would be the lab strain surrounded by this green box that people are familiar with. So Queensland has a slightly different leaf morphology. Perhaps you can see the wings to the petioles, that the flowers are bigger, that the flowers have a structure that favors outbreeding and insect pollination, whereas the lab strain is a self-pollinator. So that's just a couple of the differences, but there's quite a range of differences. Just to give you a little reminder of the evolution or the predicted evolution of what's been going on. So in tobacco, you had two diploids, Tomentosiformis and Silvestris, that about 200,000 years ago hybridized to form this allotetraploid with 12 plus 12 chromosome pairs, making 24 chromosomes. But in Benthamiana, we don't know what the parents really were, but we assume it's a diploid, Nicotiana, crossed with something else, which may be Silvestris from the chloroplast genome. We think it might be the maternal partner, might be Silvestris, but these hybridized perhaps 6 million years ago. And we think this because if you look at this clade here, these are all Australian Nicotianas, and they're all tetraploids. And then if you date them back, this point here is about 5 or 6 million years ago. So about that sort of time, you form the first allotetraploid, and then over that 6 million years, it's lost 5 of the chromosome pairs to get you down to 19 chromosome pairs. So I should say that we chose Queensland as well to study compared to the lab because they're almost the furthest apart. So when we, this one sentence summarizes several, an awful lot of effort, but I did pack biosequencing, assembled them using BioNano optical mapping, polished the sequences with Illumina, and then used high-C analysis to refine this. So we've now got the 19 chromosomes for lab and the 19 chromosomes from Queensland. This is the high-C output. And so we've annotated that genome, and it has a Busco score of 95.7, which is pretty good, about 52,000 genes we're thinking. And we've annotated it based on an orthoset of these different Nicotianas, Tobacum, Silvestris, Attenuata, Tophora, Tomentosiformis, and also Tomato and Arabidopsis. And we get, as I say, about 52,000 for the lab and 61,000 genes for the Queensland. And these numbers seem pretty good considering that tobacco is this 24 chromosome allotetraploid with no chromosome loss of about 70,000 genes, whereas these diploids range between 30 and 35,000. So this sort of fits quite nicely with a duplication, a tetraploid, but with loss. And an interesting thing that we found that if you, from basically 3 different ways of looking at it, that we calculate that about 25% of the genes from either using the Busco or looking at an RNAi pathway, just the candidates manually, or the whole genome, about 25% of the genes, they have lost one of the homologous pairs of the one member of the homologous pair. So 75% retain both, but 25% have lost them. So when we did the sequencing, we could see it comes out in different numbers based on length and various things. But what we can do is we used synthetic blocks of genes, and we can see, for example, Queensland chromosome 1 was actually the same as, or very similar to chromosome 13 in lab, and 2 with 11 and so on. So we renumbered the chromosomes so that the lab strain goes from chromosome 1 to 19 and the Queensland 1 to 19. And then you can see this synteny across the chromosomes, and we flip them around so they're all in the same order. And what you may also notice is that you can see these synthetic blocks sort of forming a line here and here, and that's the subgenome. So the way we've numbered it is that chromosome 1 in lab is actually the homologous pair with chromosome 11 and 2 with 12 and 3 with 13 and so on. And so when we take that and we compare it with tobacco, we've taken the liberty of rearranging the chromosome numbers from tobacco so that we've put all the Sylvestris subgenome here and the Tomentosiformis subgenome here. And again, you can see, hopefully, that these synthetic blocks that across each chromosome with the lab strain. And you can see the homologous chromosome pairs here. Furthermore, you can look at attenuata and, excuse me, that you can also see these lines here of synteny with the two subgenomes. And to summarize this, what we did is we took the interactions, we did a matrix of basically the detection of the orthologous genes and how many there were on each chromosome and so on, and used a network clustering analysis. And so this is this output here. And hopefully you can see that there's a cluster here, which this is the Queensland strain, but we get the same result with the lab strain. But here's Queensland chromosome 3 coming closely with Queensland 13, and it's also close to tobacco 16 and tobacco 12 and attenuata 4. Whereas here it's Queensland 16 and 6 with tobacco 4 and 6 and attenuata 3. And so we have these different clusters, which if we take this matrix of network of I guess, similarities, and just focus on just the genome, and this is the Queensland genome, but again, it's the same as that. You get this nice pairing of chromosome 1 with 11, 7 with 17, 6 with 16, etc. I guess the numbers are not surprising because it's a sort of a self-fulfilling prophecy because we've numbered them that way. The point really to point out is that chromosome 9 and chromosome 10 and chromosome 19 are the orphans. They don't seem to have a homologous chromosome. And so if we take these pairings and we can divide them into those subgenomes, we can find, if you like, the blue subgenome here, and so chromosome 1 of this subgenome is very close to chromosome 12 of attenuata, whereas chromosome 11 is close to chromosome 12 of attenuata. And if you look at this subgenome, it has large synthetic blocks with each of the chromosomes of attenuata, the diploid, but it's missing a large block with chromosome 10. Similarly, if you look at the other subgenome, it has a lot of these synthetic blocks and you can almost find the partner, but it's missing for chromosome 4, 5, 6, and 8. And so it's tempting to say that chromosome 10, 4, 5, and 6, so they represent the 5 chromosomes that have been lost when going from 24 to 19 chromosomes. I know it's a gross simplification, but it's
Peter,
nice thought.
Excuse me, you have 2 minutes to wrap up.
Okay, oops, wow, quick. So this is the summary of that. And so we have the lab strain of chromosomes 1 to 10, 11 to 19, and you can see the homology. And this is just the pairing with similarities with tobacco, so Tomentosiformis and Sylvestris. And you can see that these ones are bordered in red, showing that they've got actually, so for example, chromosome 1 is more similar to attenuata 12 and 24 than chromosome 11 is to 12 and 24. So we're able to separate them out into the subgenomes based on the similarity to attenuata and the tobaccos. So what we've done is we've put these up on a web browser, and that we're going to release this just to the public on the 24th of November, which happens to be my birthday, so that's when we decided we would release it. And if you just go to bent.com, you'll find that the chromosome level reference sequence and some RNA profiles and the masking of the transposons. And so just to quickly give you a quick insight, here's just some genes that people use as reporters and stuff, and so they're just mapped onto the different chromosomes. And to give you one final look is that this RDR1 is a gene that we were very interested in that lab strain was defective in this gene, and we thought this is what has given the lab strain a particular ability to express foreign genes and not be able to fight off viruses. And we were interested to see if this was a general trait, that the lab strain had lost a lot of these, a lot of pathways that it didn't need in an effort to be more vigorous. And here's just an example. So in Queensland, it has those large flowers and it produces this benzyl alcohol at night to attract the moth, whereas lab doesn't, it's a self-pollinator. And the downside of producing this moth-attracting compound, volatile, is that you may attract herbivores that will eat you. And so Queensland actually produces high levels of nornicotine, which is much more potent than nicotine. And Queensland produces this, whereas lab is dysfunctional in this gene that demethylates nicotine to nornicotine, and so doesn't produce that. And then finally, just another gene a pathway that seems defective in the lab strain is the ability to produce anthocyanin. If we fish for the MIBs that induce the anthocyanin pathway, and there are 2 of them, and in the wild strain from Northern Territory, if you squirt these into the leaf, they can produce the anthocyanin, induce anthocyanin, whereas in the lab, neither of these genes is functional. In Queensland, there's only one. And lab is also dysfunctional, it seems, in its ability to produce to store anthocyanin. If you induce it in the lab, I don't know what's happening here, but this is a patch of death. It should be green all around, and that lab is unable to, we think, put it into the vacuole, and so it actually kills the plant. And so I hope that's given an insight into the lab strain and the Queensland strain, and that this will be of use to researchers in both academia and in biotech industry in studying their pathways. And at that point, I'll finish and answer any questions if there are any.
Do we have time for just one question? Please use the Q&A. Says Peter, interesting presentation. The duplication and specialization of the chromosomes made independent chromosomes. Besides specialization, are there other mechanisms to avoid recombinations?
So I think the answer is that we see there's a whole lot of recombination that's going on. And that when I say that you can detect the 2 homologous chromosomes, it's probably about 30–40% that you can see in matching in tobacco or the attenuator or commercial tobacco. And that there is a lot of recombination going on, it would seem, between the homologous chromosome pairs. They're not actually totally immune to crossing over between them in that 6 million years.
Okay. All right. Okay. Thank you, Peter. And again, you can catch up with Peter if you have other questions in the Zoom meeting room later. And we're going to move on to our next speaker.
Okay. And the next speaker is Johan Hunziker, University of Tsukuba, and his title is Improvement of Carotenoid Accumulation in Tomato Fruits by CRISPR-Cas9 cited in the aminase system.
Thank you very much. So, sharing my screen. I still need to get Mr. Waterhouse to remove his sharing screen.
Oh, sorry, am I still sharing?
Yeah.
And you have to make Johan the host.
Stopping share, sorry. Is that better?
Yes. Is it fine? Is it fine or not now?
Yeah, it is.
Okay, so let's start. Thank you very much for giving me this opportunity to make this talk today. So my name is João Anziker, and I'm a PhD candidate under the supervision of Professor Ezura at the University of Tsukuba, Japan. And today I will introduce you my PhD research about the accumulation, the improvement of accumulation of carotenoid in tomato using a base editing system, which is CRISPR-Cas9 fused with a cytidine deaminase. And for this, we select tomato because of his as a plant model for fleshy fruits, and more especially for his high accumulation of carotenoid, and more especially lycopene and beta-carotene in the mature fruits. And as one of the most consumed crops in the world, we are focusing this aspect for this technology for trying to improve this accumulation. So for this, our target was the creation of high carotenoid accumulant tomato fruits mutant by using a multiple target approach by this technology, which is also called target AID system. So a brief introduction of what is target AID. This is Cas9, which is fused with a cytidine deaminase. So the concept is to use the ability of Cas9 to target a specific portion of the genome with the guide RNA. And we used nickase 1 in order to reduce the phenomena of insertion deletion. And our cytidine deaminase is inducing a substitution of cytidine by a uracil, and during the duplication of DNA, we could get a substitution to a thymine. And this has been already reported in bacteria and also in plants such as rice and tomato, but until now only on single target. So our objective was, by using this tool of genome editing, to create new allelic version of multiple targets which are simultaneously transformed in a single generation. And for this, we focus our attention on improving the accumulation of lycopene more particularly. So we divide our project in two parts. One, which was analyzing the efficiency of multiple transformation in one time, in one generation, one plant. And the second, of course, is a characterization of this newly generated allele for more especially the carotenoid accumulation, but also the other impact on the plant. So for this, we select three targeted, which was well described in the literature the past 2 decades, and more especially the DDB1 and DET1 protein, which are also known as HP1 and HP2 mutants. And the third one, which is a lycopene beta cyclase, the chromoplastic version, which is also known as OGC mutation, Olgot crimson mutation. And we select more especially our target, which was close to the original HP1 and HP2 mutation allele. And for OGC, we select one which was expecting to create a stop codon by substitution. So a brief reminder of what is DDB1 and DET1. There are two proteins which are working in a CDD complex on the light signal, and more especially in mutation in these both proteins, which induce usually an increase of chloroplast content, which after maturation of the fruits induce an increase of chromoplast amount. So which means we don't directly target the carotenoid pathway, but more increase the number of chloroplasts in the fruits. Our CYC mutation is just to disrupt the catabolism of the lycopene in beta-carotene, and so by this way, just increase and blocks the carotenoid pathway in lycopene. So for this, we perform a traditional agrobacterium transformation and collect the T1 seeds that we segregate, and in order to only isolate null segregant line in order to prevent any somatic effect of mutation of remaining activity of Cas9. And then we segregate them in order to purify each line in a homozygote condition, and also we backcross with a wild type in order to isolate each individual allele we could create it. And by this, after we analyze the plant development and also the fruit carotenoid accumulation. So we perform an NGS sequencing to amplicon sequencing to analyze efficiency of transformation for each target. And as expected, we had a substitution of cytidine by a thymine for both targets, and more especially in between 15 and 20 base pair from the PAM sequence, as it has been reported previously by Nishida group. And our efficiency of transformation was quite high, but we had between 50 to almost 100% of substitution. And the further from the PAM sequence we was, and higher was the efficiency. And we got almost all plants regenerated with a triple substitution simultaneously. So we got several patterns of substitution. And for DDB1, we got two patterns, which was a single substitution and a double substitution. For that one, we got a triple substitution, a double substitution, one single here, which was a silent mutation, and then another one, which was a quadruple substitution, but was very, very few amount of plants with this mutation. And for CYC, we got two patterns, which was a triple substitution and a double one, but both of them was inducing a stop codon as expecting. So we focused our attention on plants which was carrying a single and double substitution in DDB1, but was also both lines showing the same pattern of mutation for DET1 and CYC. So here is our aspect of triple plants, triple mutated plants. So we have these two one which are triple with a single substitution on DDB1. The last one, which is a double substituted homozygote for DDB1, and the middle one, which was heterozygote for DDB1. And what we notice is we have plants which are smaller compared to the wild type and with darker green fruits. So we observed the plant development, and what we could get is, as expecting from the mutation on DDB1 and DET1, we got a dwarfer plant for triple mutant, but also for double mutants, we got a dwarfer plant compared to the wild type. And for our double substituted DDB1 in triple mutated background, we got even a much more dwarfer plant. And this also was reported on the fruit development. But on the opposite, our triple mutant didn't show a significant impact on the fruit development, and our single mutant also didn't show it. So this double substitution in DDB1 looks to be too much impacting the plant development, so we don't really keep this for further analysis. So for chlorophyll accumulation, same, our mutant in DDB1 and DET1 was reported to show an increase of more especially total chlorophyll amount that we can see in the black bar. So our triple mutant single SNP was showing a much higher accumulation of chlorophyll, which was also same reported in the double mutant DDB1 DET1. And our DET1 single mutant was also showing a darker color, but we can also see more or less on the pictures. And interestingly, our DDB1
2 minutes to wrap up. 2 minutes.
Sorry?
To wrap up.
Okay.
2 minutes.
Okay, so we didn't get a significant difference for DDB1. So, but we still get an addition of effect for the double mutants. For the fruit color, we didn't observe a significant difference in the color. So we moved to the carotenoid accumulation, and what we notice, we have a significant increase in lycopene accumulation for all mutants, except the double substituted one. But interestingly, we still also observe an increase in beta-carotene accumulation, which was not expecting, as we was thinking to disrupt this lycopene beta cyclase. So we cannot really explain this issue except of we have a remaining activity, but we couldn't observe it until now. So to summarize, our technology could help to the generation of simultaneous substitution in different targets in a single generation. And our targeted gene was altered, and our plant shape also. We could increase our carotenoid accumulation as expecting. And surprisingly, we didn't get a phenotype expecting of disruption of the lycopene beta cyclase. So this still remains unclear. And we couldn't also observe the same phenotype of darker flower regarding this. So here is our partner. We would like to thank more especially the University of Kobe with the Professor Nishida and Professor Kondo, who provide us this vector for this study, and the Tomatoma project, which providing us material support for plants, the Ministry of Education and Sports, which provide me this financial support for the PhD, and the SIP project, which was funding us for all this project in collaboration with the government. So this is the end of my talk. If you have any question, please feel free to ask me or send me an email later. I'm not sure if I can participate to the talk show because it's a bit late here in Japan, but feel free to send me an email.
Thanks, Johan. We already have 3 questions.
Okay.
The first one is from Flavio Joao Soares.
Okay.
Presentation, Johan, did you check the expression of the targeted genes?
I checked them, yes. I don't have the data on the slide. The expression was increased. Expression of DDB1 and DET1 was increased.
Okay, good.
Photosynthetic parameter, unfortunately, we didn't analyze it yet.
One question from one question from Magdalena Rossi. Have you analyzed any photosynthetic parameter, chlorophyll fluorescence, for example?
We just measure the chlorophyll content until now in fruits.
And how is it?
I show the slide, we increased the chlorophyll amount.
Okay, then a third one from Matteo Martina. Thanks for your presentation. Did you by chance investigate the carotenoid volatiles accumulation in your mutants?
No, we didn't analyze the volatiles. We just focus our attention in the lycopene, more especially in the lycopene accumulation in the fruits.
Are you thinking to do it?
I will ask to my professor if it's planned or not, but for the moment, not yet. It was not.
And one last question from Jessica Cooperstone. This might have been something I missed, but what background variety was this work done in?
It was done in the microtome background.
Microtome, okay.
Microtome background.
And one more, Lucio D'Andrea. I was wondering if you have checked the impact of these mutations in plastic number or structure.
Plastic structure, no, but number, we are doing it now.
Okay, that's great. That was great timing. Lots of questions answered. Thank you very much. And I leave the floor to Joyce.
Thank you very much for your question.
All right, if you could stop sharing your screen, please.
Yes.
Our next speaker is Eva Nock. Eva was at Riken in Japan. However, she is currently at the Gregor Mendel Institute in Vienna. And she's going to tell us about her work that she did when she was in Japan with analyte biosynthesis. Hi.
Okay. Thank you, Joyce. I'm just trying to share my screen.
Okay, I can see your screen.
Is it working?
It's working.
Perfect.
Great.
Good. Great. I had a little bit internet problems, but I hope it will work out through this presentation. Okay. I have to go here.
Looks good.
Good. Okay. Yeah, thanks for the introduction. As Joyce said, this is work I did in Riken in Japan while I was doing a postdoc there. It's about methanolite biosynthesis. And just a little brief slide. Methanolites, they are a diverse group of steroidal lactones. So they are based on this backbone here. And then throughout the Solanaceae, there are different modified versions of this methanolite backbone. So there are lots of different structures in different plants. And the methanolites, they have their name from Methania, somnifera, which is this plant here. It's used in the Indian traditional medicine. It's called ashwagandha, if somebody has heard this before. So they have some anti-inflammatory and antibacterial properties. These Vidhanolites. So the Vidhanolites are not just produced in Vidania, but in several plants of the Solanaceae, mostly from the Solanoidae subfamily. So here is basically, again, the core structure from Vidhanolites from Vidania. Then in Physalis, and especially in Physalis alkekengi, you have these very complex physalins, which have this cage structure and a lot of modifications. And on the tree on the right here, you can see it's a phylogenetic tree of some of the Solanaceae, and especially the Solanoidae here. And I've marked with the blue dot those tribes where Vidhanolites have been described in literature. So yeah, lots of different plants make these Vidhanolites. And we were especially interested in the physalins because of their complex chemicals, so the complex structures. And as shown here, so Physalis alkekengi is this very decorative plant, has these complex physalins, and Physalis peruviana, which is the edible kind, makes more simple compared to the physalins Vidhanolites. So we wanted to identify some of the genes responsible for these complex structures of the physalins, and did an RNA-seq, so comparative transcriptomics approach, which is published in Frontiers, and the data is available. So if anybody is interested in Physalis transcriptomes, you can find them. So we did RNA-seq analysis and then looked for differentially expressed genes, and we're hoping to identify candidates for these physalin biosynthesis. Unfortunately, that didn't work out quite as we had planned, but instead we found a very interesting gene candidate. And this is a dwarf 1 homolog, which we named 24-iso, and this is what I will talk about now. So this, the Vidhanolite biosynthesis based on isotope tracer experiments. We know that both 24-methylene cholesterol and 24-methyl-desmosterols are precursors for Vidhanolites, and our hypothesis is that then the biosynthesis goes in this direction, where then the double bond from 24-methylene cholesterol is moved from the 24-28 to 24-25, and then further steps happen to the Vidhanolite scaffold. So because we have this double bond here in the final Vidhanolites. Basically this is then the reaction we're looking at, and somehow my timing is running. So we know this reaction already. So dwarf 1, or also steryl side chain reductase 1, is an enzyme that does a 2-step reaction. So it does the isomerization and then a reduction of this bond. And then there's the steryl side chain reductase. So this, dwarf 1 is involved in the brassonolite biosynthesis pathway. Then the steryl side chain reductase 2, only found in Solanaceae, and it's involved in cholesterol biosynthesis, and it catalyzes the reduction of the 24-25 bond. And this is a homolog of dwarf 1. So we found this third homolog in Vidania and in our Physalis transcriptomes, and we thought that this might then be the 24 isomerase that isomerizes this double bond here. So looking back at this slide here again, I looked at basically all the ones here that are underlined. We have transcriptome or genome data available, so I checked for the presence of the 24-iso gene, and it fits very nicely with those tribes where we also find Vidhanolites described. So there's an overlap. And then looking at the phylogeny of the dwarf 1 homologs, as I mentioned, so there's the dwarf 1, which is basically in all land plants, and it's involved in brassonolite biosynthesis. All the Solanaceae plants have steryl side chain reductase 2, which is involved in the cholesterol biosynthesis. And then in the Solanoidae, mostly in the Solanoidae subfamily, we find the 24-iso gene. yeah, and so basically this is showing in the same thing in a different way, that we have one gene duplication here, which leads to the SSR1 and SSR2, and then another one that leads to the 25, so in the Solanaceae. So to determine the activity, we cloned the genes from 4 different species. So we have Withania, Physalis, chili, and also Petunia, and then did expression in tobacco and also in yeast. And I'll start by showing the yeast data. So for this, I used a yeast that has a modified sterile biosynthesis. So it produces 24-methylene cholesterol. So this is the empty vector control here. You can see the yeast strain produces 24-methylene cholesterol, which we think is the substrate for the 24-iso enzyme. Then yeast expressing the 24-iso, we can detect the 24-methyl-desmosterol, and then this is the 24-methyl-desmosterol standard. So we can actually see in yeast that the 24-iso does what we expected it to do. And we also did in vitro activity assay with protein purified from the yeast. And so we saw that this reaction requires NADPH. So without NADPH, we get no production of 24-methyl-desmosterol. Then for the expression in tobacco, just to confirm that it's also functioning in plants. As you can see, the top trace here, this is a GCMS traces. Again, the top trace is the empty vector control, and this is the pink one is the extracted ion chromatogram. And you can see here that normally tobacco produces little free 24-methylene cholesterol. There's a tiny peak. But then when we transform 24-iso in the tobacco leaves, this peak disappears, and instead we have a tiny 24-methyl-desmosterol peak here. So also in plant, we could show that 24-iso produces 24-methyl-desmosterol. And just to confirm, we also did subcellular localization. And so sterile biosynthesis happens in the ER, and our 24-iso also localizes to the ER in tobacco. Then to show that 24-iso is really involved in withanolide biosynthesis, we used VIX to silence this in Withania plants. And you can see here on the picture, this is Withania plants with the PDS positive control. So the white is the silencing. And we used the tobacco rattle virus vector system for this. But there was a problem. So because the SSR1, 2, and the 24-iso, the gene sequences are very, very similar. And we couldn't really find a unique stretch that was unique to the 24-iso. So tried several different constructs, and both 24-iso and the SSR1 or dwarf1, they were always silenced together. So instead we came up with a solution. Instead of taking one stretch, we took 3 different unique regions from 24-iso and combined those together in the TRV2 vector. And then looking at gene expression in the silenced plants, the bars are the 24-iso silenced plants, and we were able to only reduce 24-iso and not the SSR1. So SSR2 was not that big of a problem because it's not expressed in the leaves of Withania. So then next that we silenced the 24-iso, we did GCMS analysis of the sterols. And the bottom trace here is the control plants that produce 24-methyl-desmosterol. And in the silenced lines, we have almost no peak here, but instead we see a big increase in 24-methylene cholesterol. So the product accumulates in these plants. Then we also did LCMS analysis.
2 minutes to wrap up, Eva.
Good, thank you. Then we also did LCMS analysis of vidaferin, and it's also reduced in our 24-iso silenced plants. So we could really show with this one that 24-iso is involved in withanolide biosynthesis. And then this is basically our conclusion slide showing here. So there's the dwarf 1, SSR1, which is involved in the brasinolide biosynthesis. Then in the Solanaceae plants, there's the sterile side chain reductase 2, which is involved in cholesterol biosynthesis. And then we have a third pathway in the solanoidal plants that produce vithanolides, where 24-iso branches off this to the vithanolide biosynthesis pathway. And then just very briefly, maybe in the last minute, there are of course open questions. So there are further biosynthetic enzymes, and then also the role for the 24-iso in Petunia. And I just skip quickly over this. But basically in Petunia, because the genome sequence is available, we identified the gene cluster with 24-iso and some cytochrome P450s and 2 OGDs. And Petunia doesn't, so vithanolides, but they have these Petunia steroids, which don't have this double bond, but they are related compounds. And it's also, so the 24-iso is expressed in Petunia plants, but we ran into a problem trying to find out what it does in Petunia. So you can see here, these are Petunia plants where we try to silence the 24-iso using VIX again. And the control plants, they look fine, but our 24-iso silenced plants, they died. So this just opens a question if the role of this enzyme is very different in Petunia than it is in the Withania plants, for example, and what role those specialized metabolites might be completely different in Petunia. And with this, I come to my end. I would like to thank all the people involved, of course, my colleagues at RIKEN, and then the Casper Foundation and the Japanese Society for Promotion of Science for funding my research stay. And thank you for listening.
All right, thank you, Eva. Do you have any questions for Eva? And please use the question and answer box.
Joyce, may I ask a question? I cannot use the box myself.
Sure.
So is cholesterol biosynthesis specific to the Solanaceae? And that's not directly. And with and how widespread phylogenetically are cholesterol biosynthesis and vithanolide biosynthesis? Do you find them in other plant taxa other than Solanaceae?
So the SSR2 is restricted to the Solanaceae, and the 24-iso I could only find in those Solanoidae plants and Petunia and Chili, which are a little bit outside.
What about the products?
The products, yeah, that's more tricky because so I think I'm not quite sure with the cholesterol. I think maybe somebody else knows better, but cholesterol is also outside the Solanaceae. So with the vithanolides, there are some corals that make some similar products. So very different, not even plants, but some soft corals also produce vithanolides, but that would be completely independent.
Glycoalkaloids.
Sorry?
I think Liliaceae make glycoalkaloids, if I'm not mistaken.
Maybe, I don't know.
We have one more question. What are these vithanolides for? And the second part of that is, can you comment a bit more on your strategy to silence the gene without silencing the other two homologs?
Yeah, so what they are for, we don't really know, I think. So yeah, there's a lot of more research that needs to be done. Maybe they are anti-fedent. So the petuniolides, they work against caterpillars. And so maybe the vithanolides are similar. They are mostly also in the roots. So it might be some against some root pathogens, but it's nothing has been shown about that yet. And the strategy to silence the genes and not the other two homologs. Yeah, so basically we tried different lengths. So we tried from 100 to 300 base pair length in the TRV2 plasmid, and we always saw that SSR1 was also silenced. So instead, I think we ended up taking around 30 to 40 base pair, these three pieces from different regions of the 25. So we did alignment and then looked for the most different parts, basically, and then put them together into the TR, so just after each other into the TRV2 plasmid. And then that worked just assigned as 25. So.
Okay, great. All right. Thank you, Eva. I'm going to turn it over to Giovanni now and for our last speaker of the session.
Okay, there's one more question on the chat. You're going to dismiss it?
Well, we're going to keep on time, and I think it's just a comment about the cholesterol pathway.
Okay, good.
Yep.
So, thanks, Joyce. So we have moved to the last presentation in this session, which is by Lorena Ramirez, Wageningen University. And the title is A New